Assalamu alaikum dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 49. The topic is the pathway of electron transport, or in other words, electron transport chain. Discussing agenda for the topic will be introduction, the pathway of electron transport, and then we will discuss chemiosmosis. Lecture outcome. After watching and listening this lecture, students will know about the electron transport chain and its various complexes and how this process leads to the formation of ATP. Introduction. During oxidative phosphorylation, chemiosmosis couples electron transport to ATP synthesis. So electron transport chain occurs in the inner lumen of mitochondria, and in this process ATP is generated by a molecular motor called ATP synthase, and the hydrogen ions are involved in the synthesis of ATP, where the hydrogen ions move inside into the inner lumen from the intermembrane space, and they passes through the molecular motor or the ATP synthase, and in this way ATP is synthesized. We will discuss this process in detail in the subsequent slides. So our main objective in this chapter is to learn how cells harvest the energy of glucose. Glucose is a 6-carbon molecule, and how the energy is derived from this 6-carbon molecule and other nutrients in food to make ATP. So how the cells synthesize ATP, which is the energy currency of the cell, which perform different functions like mechanical work and chemical work. So how this molecule is synthesized. But the metabolic components of respiration we have examined so far: glycolysis, in which a 6-carbon glucose is broken down into 2 molecules of pyruvic acid, each having 3 carbon, and then pyruvic acid is converted into acetyl coenzyme A, which enters into the Krebs cycle or the citric acid cycle. So in these 2 steps, only 4 ATP molecules are produced by glucose. Remember, 2 net ATPs in glucose, and then 1 ATP for each pyruvate molecule that enters into Krebs cycle. So a total of 4 ATPs are produced, and remember, these 4 ATPs are produced through substrate level phosphorylation, in which one of the intermediates, one of the substrate of the metabolic pathway, provides a phosphate group to ADP and produces ATP. So these 4 ATP molecules, 2 in glycolysis and then 2 in citric acid cycle, are produced through substrate level phosphorylation. Now we just discussed the production of ATPs, but alongside ATPs, high energy electron carriers like NADH and FADH2 are also produced in glycolysis, then in the conversion of pyruvate to acetyl coenzyme A, and then further in the Krebs cycle. So what about these NADH and FADH2 molecules produced? How they will provide more energy, how they will help in the synthesis of more ATP molecules. So at this point, molecules of NADH and FADH2 account for most of the energy extracted from each glucose molecule. So the high energy electron carriers were accepted by NAD and FAD, and as a result they become reduced. Now they are having high energy electrons, and this high energy electrons will be delivered to the electron transport chain, where more ATPs will be produced. Perhaps or the major bulk of ATPs that is produced in aerobic cellular respiration is through oxidative phosphorylation, which takes place during electron transport chain, and these are the high energy electron carriers which start this process. These electron escorts link glycolysis and citric acid cycle, because these are the products of glycolysis and citric acid cycle. So they will run the machinery which will result in the production of ATPs through oxidative phosphorylation, which uses energy released by electron transport chain to power ATP synthesis. In this section, you will learn first how the electron transport chain works, and then how electrons flow down the chain is coupled to ATP synthesis. So the electron transport chain is basically composed of 4 complexes where different carriers of electrons are present, and these electrons are passed on from one carrier to another carrier. There are a series of reduction and oxidation reactions, and in this way, so how this process leads to the synthesis of ATPs, we will discover soon. The electron transport chain is a collection of molecules embedded in the inner membrane of the mitochondria in eukaryotic cells. So if you remember from the mitochondria topic, mitochondria has 2 membranes. It's a double membrane structure. The outer membrane is smoother. The inner membrane is thrown into several enfoldings which are called cristae, and these enfoldings are to increase the surface area. For which purpose? Because these inner membrane has those molecules in hundreds of thousands of number. So these molecules are they houses the electron carriers. So these enfoldings increase the surface area, and as a result, a lot of complexes can be there. The foldings of the inner membrane to form cristae increases its surface area, providing space for thousands of copies of each component. So the more surface area, then there will be room for more complexes to accommodate. Now here is yet another example of structure determines the function. So these enfoldings are to increase the surface area, and if the surface area is more, then there will be more spaces for complexes to reside into. The enfolded membranes with its concentration of electron carrier molecules is well suited for the series of sequential redox reactions. So there are a series of oxidation reduction reactions. Most components of the chain are proteins, which exist in multi-protein complexes numbered 1 through 4. So this is the first complex, the second complex, the third complex, and the fourth complex. In some of the books you will also find a fifth complex, which is the ATP synthase, but here in the book we are following, they have only mentioned 4 complexes. Now you can see here each complex has carriers of electrons, and these are lying in a sequence, and there is a series of redox reactions. A carrier molecule is first reduced and then is oxidized when it passes the electrons to another carrier. And finally, oxygen is the final acceptor of electrons. Oxygen is a highly electronegative atom. So this process is known as aerobic cellular respiration because of this, because here oxygen acts as the final acceptor of electrons. So the oxygen which you inhale is basically used here. Tightly bound to these proteins are prosthetic groups. Now they are the multi-protein complexes, and to those multi-protein complexes are attached prosthetic groups. What is a prosthetic group? Prosthetic group is a tightly bound, specific, non-polypeptide unit, and it is required for the biological function of some proteins. Now, the prosthetic group may be an organic molecule such as vitamin, sugar, or lipid, or it may be an inorganic molecule such as metal iron, but it is not composed of amino acids, means it is not a protein in nature. Now here we have two more terms, cofactors and coenzymes. What is a coenzyme? Coenzymes are organic molecules, and they quite often are bind to the active site of an enzyme, and it helps in the substrate recruitment. Whereas on the other hand, the cofactors, they do not bind the enzyme. Cofactors are helper molecules and can be inorganic or organic in nature. So basically these are the two groups of molecules for the proper functioning of proteins. Now, figure 1 shows the sequence of electron carriers in the electron transport chain. Why it is known as electron transport chain? Because electrons are transferred from one carrier to another carrier. And during this electron transport, electron carriers alternate between reduced and oxidized state. So first, a carrier is reduced by accepting electrons, and then when it delivers its electrons to another carrier, then it becomes oxidized. So that's why in electron transport chain there are a series of oxidation reduction reactions. Each component of the chain becomes reduced when it accepts electrons from its uphill neighbor, which has a lower affinity for electrons. So this has a lower affinity, this one has higher affinity, this one has even higher. So as the carriers, as you move down the chain, the electronegativity of the carriers increases. And then finally the oxygen arrives, which is the most electronegative atom in this group of carriers. It then returns to its oxidized state. So a carrier is first reduced and then oxidized when it passes electrons to its downhill neighbor, which has a higher affinity for electrons. So first reduced when passes electrons to next carrier, then oxidized, and this is the order of ascending order of electronegativity. Now let's take a closer look at the electrons as they drop in energy level passing through the components of the electron transport chain. So now we will discuss each carrier. We will first look at the passage of electrons through complex 1 in some details as an illustration of the general principle involved in electron transport chain. So from where these electrons will come? Okay, we accept that these are the carriers of electrons and they pass electrons from one to another, but from where these electrons come? Remember those NAD and FADH2 which are produced in glycolysis, in link reaction, and then in Krebs cycle. So these high energy electron carriers NADH and FADH2, actually these are the ones which starts this chain. These are the ones which deliver their electrons and start this chain. So electrons acquired from glucose by NAD during glycolysis and the acetic acid cycle are transferred from NADH to the first molecule of the electron transport chain in complex 1. Now remember one thing, that in the first two stages there were two different types of electron carriers. The first one was NADH and then in Krebs cycle it was FADH2. So remember that NADH delivers its electrons to complex 1, whereas FADH2, it passes the first complex and it delivers electrons to complex 2. NADH complex 1 and FADH2 complex 2. So that's why when NADH is a source of electrons, then more ATPs are produced as compared to FADH2. Now, which is the first acceptor of electrons? Suppose NADH has arrived in complex 1 and it has donated its pair of electrons. Now, FMN is the first acceptor of electrons, flavoprotein, so named because it has a prosthetic group called flavine mononucleotide. So in electron transport chain, this is the first acceptor of electrons. The next acceptor of electrons is iron sulfur protein. The third acceptor of electrons in electron transport chain is ubiquinone. This electron carrier is a small hydrophobic molecule and it is the only member of the electron transport chain that is not a protein. Only member in sense of carrying electrons, so it is the only electron carrier which is not a protein. Now ubiquinone is individually mobile within the membrane rather than residing in a particular complex. So another name for ubiquinone is coenzyme Q or CoQ. Now most of the remaining electron carriers between ubiquinone and oxygen are proteins called cytochrome. So the first one is flavoprotein, the second one is iron sulfide, the third one is ubiquinone, and thereafter you can see there are a series of cytochromes with different names and all of them are proteins. Their prosthetic group called a heme group. They are talking about cytochromes. They have a prosthetic group which is a heme group and it has an iron atom that accept and donate electrons. Now the heme group is a cytochrome is similar to the heme group in hemoglobin. Now the heme group of cytochrome is similar to the heme group of hemoglobin, but what is the difference? The difference is that the protein of red blood cell transport oxygen, whereas the heme group of cytochrome transport electrons. The electron transport chain has several types of cytochromes, each named CYT with a letter and number to distinguish it as a different protein with a slight different electron carrying heme group. So you can see here cytochrome B, and then you have cytochrome C1, cytochrome C, then you have cytochrome A, cytochrome A3, and finally the electrons are accepted by oxygen, and at the same time oxygen binds with two hydrogen molecules and form water. The last cytochrome of the chain CYTA3 passes its electrons to oxygen in O2, which is very electronegative. I hope you remember the electronegativity, which is the ability of an atom to attract a pair of electrons towards itself. So oxygen has high electronegativity, and it readily accepts a pair of electrons. Each oxygen also picks up a pair of hydrogen ions, protons, from the aqueous solution, neutralizing the -2 charge of the added electrons and forming water. So this is the end product. Water is formed, and this water is known as metabolic water. Another source of electrons for the electron transport chain is FADH2. FADH2 is produced in Krebs cycle and it enters complex 2. Remember, not complex 1. So it has lesser ability to synthesize ATPs as compared to NADH. Means when it participates in electron transport chain, lesser ATPs are produced as compared to NADH. Notice in figure 1 that FADH2 adds its electrons from within complex 2 at a lower energy level than NADH does. Consequently, although NADH and FADH2 each donate an equivalent number of electrons, that is 2 for oxygen reduction, they both of them donate 2 electrons, but because of their entry, NADH enters complex 1, FADH2 enters complex 2. That's why their ability to help in the synthesis of ATP is different. Chain provides about 1/3 less energy for ATP synthesis when the electron donor is FADH2 rather than NADH2. Why? Because of their entry into the chain. We will see why in the next section. When we discuss chemiosmosis, then we will see why the FADH2 molecules helps in the production of lesser amount of ATPs as compared to NADH. Now the electron transport chain makes no ATP molecules. So now basically we are discussing the production of ATP molecules through oxidative phosphorylation, which takes place in the electron transport chain. But so far we have only discussed the complexes and the carriers of electrons and the oxidation reduction reactions and the final acceptor of electrons. So far there is no sign of production of ATPs. So where those ATPs are produced? Instead, it eases the fall of electrons from food to oxygen, breaking a large free energy drop into a series of smaller steps that release energy in manageable amounts. Now, what is the significance of electron transport chain? Because the ATP molecules are produced slowly and gradually, and they are produced in such a way which is more feasible for the cell. Now, remember from the second law of thermodynamics that during every energy transformation, some part of the energy is wasted as heat, and this wasted energy is unable to perform work. So similar is the case here. If all the energy present in glucose molecule is yielded at once, then a large amount of this energy will be wasted as heat. Therefore, these several steps are designed with an aim to derive maximum benefit, maximum usable ATPs from glucose. How does the mitochondria or the plasma membrane in prokaryotes? Now remember that prokaryotes lack mitochondria, but these complexes are embedded in the plasma membrane of prokaryotes. So this process can also occur to a certain extent in prokaryotes. The electron transport chain can also occur in prokaryotes to a certain extent, but it is not as efficient as if it occurs in mitochondria, because mitochondria is the suitable destination for this process because of the availability of all the enzymes. Now how ATPs are produced? The answer is chemiosmosis. Now what actually happens? Populating the inner membrane of the mitochondria or the prokaryotic plasma membrane are many copies of a protein complex called ATP synthase. This is the enzyme, this is the molecular motor which is responsible for the synthesis of ATPs. The enzyme that makes ATP from ADP and inorganic phosphate PI. So how does this process work? Now how does this process works? There are ion pumps which are present in the inner membrane, and as the electrons are passed from one carrier to another carrier, in this process energy is released, and this energy is used to draw the electrons through these channels into the intermembrane space. Enzymes can catalyze a reaction in either direction depending on the delta G. Delta G is the free energy. Free energy is the energy which can perform work. So those reactions which are exergonic, which doesn't require the input of energy, will be more feasible for the enzymes to catalyze. Under the conditions of cellular respiration, rather than hydrolyzing ATP to pump protons against their concentration gradient, ATP synthase uses the energy of an existing ion gradient to power ATP synthesis. Now this is the inner lumen of the mitochondria. Here are present lot of hydrogen ions, and as the electrons are passed from one carrier to another carrier to another carrier, in this way energy is liberated, and this energy is used to draw or to force or to pump the hydrogen ions from the inner lumen into the intermembrane space. Remember which energy is used? The energy which is produced in oxidation reduction reactions is used to pump the hydrogen ions into the intermembrane space. Now there is bulk of hydrogen ions in the intermembrane space, and they will move here and there, and they will try to find a way to sneak into the inner lumen again, and all of a sudden they will find the ATP synthase. ATP synthase will provide them the route through which they will move into the inner lumen. And in this way, when these hydrogen ions move through ATP synthase, this molecular machine produces ATP. So the ATP, the power source for ATP synthase is a difference in the concentration of hydrogen ions on opposite side of the inner mitochondrial membrane. Remember, this is the intermembrane space, and because of those oxidation reduction reactions, lots of ions are pumped into intermembrane space, and here they possess potential energy. There is a concentration gradient, and then they move, they try to sneak inside the intermembrane space, and they find the molecular motor through which they move inside. This process, in which energy stored in the form of hydrogen ions gradient, energy stored in the form of hydrogen ion gradient across a membrane is used to derive cellular work, such as the synthesis of ATP, is called chemiosmosis. So this process is called chemiosmosis. They are there in the bulk, they possess the potential energy, and now they will move along the concentration gradient inside into the inner lumen, and all they find will be a molecular motor or ATP synthase. They will move through this molecular motor, and in this process this molecular motor will rotate, and ATP synthesis will take place. The word osmosis was previously used in discussing water transport, but here it refers to the flow of hydrogen ions across a membrane. So now you can see here, this is the intermembrane space where the hydrogen ions are present in bulk, and then we have the molecular motor, which has 4 different components. This one is the stator, this is the rotor, this is the internal rod, and finally the catalytic knob, which catalyzes the synthesis of ATPs. So first the electrons, first the hydrogen ions, they enter into this stator and they bind to different sites, and the hydrogen ions enter binding sites within a rotator. These are the binding sites, changing the shape of each subunit so that the rotor spins within the membrane. So once these hydrogen ions binds to these sites, then this rotor starts spinning. Each hydrogen ion makes one complete turn before leaving the rotor and passing through a second channel in the stator. So first they enter here, then they take a full turn, and then they come inside into the inner lumen through this route. And when this rotor starts spinning, energy is supplied via the internal rod to the catalytic knob, where the catalytic knob also starts spinning, and in this way ATP is synthesized. Spinning of the rotor causes an internal rod to spin as well. This rod extends like a stalk into the knob below it, which is held stationary by part of the stator. It is held stationary because some part of the stator is attached with it, but because the spinning has started in the rotor region, and this energy of spinning will be transferred through internal rod to the catalytic knob. The catalytic knob will also start rotating or spinning, and in this way ATPs will be produced. From studying the structure of ATP synthase, scientists have learned how the flow of hydrogen ions through this enzyme powers ATP generation. It is just like a turbine where the water flows from higher level, and they help the spinning of the turbine, and in this way electricity is produced. ATP synthase is a multi-subunit complex with 4 main parts, each made up of multi-polypeptides. 4 main parts are stator, rotor, internal rod, and catalytic knob. Protons move one by one into binding sites on the rotor, causing it to spin. That catalyzes ATP production from ADP and inorganic phosphate. The flow of protons thus behaves somewhat like a rushing stream that turns a water wheel. ATP synthase is the smallest molecular rotary motor known in nature. How does the inner mitochondrial membrane or the prokaryote plasma membrane, remember that electron transport chain in prokaryotes occurs in plasma membrane, generate and maintain the hydrogen ions gradient that drives ATP synthesis by the ATP synthase protein complex? So from where this energy is supplied, which helps in the pushing of hydrogen ions from the inner lumen to the intermembrane space, from where this energy is provided. Establishing the hydrogen gradient is a major function of the electron transport chain, which is shown in its mitochondrial location in figure 3. Now the chain is an energy converter that uses the exergonic flow of electrons from NADH and FADH to pump hydrogen across the membrane from the mitochondrial matrix into the intermembrane space. Now the flow of electrons from one carrier to another carrier, this energy is utilized for pushing the hydrogen ions from the internal matrix to the intermembrane space. The hydrogen has a tendency to move back across the membrane, diffusing down its gradient, and the ATP synthase are the only sites that provide a route through the membrane for hydrogen ions. As we described previously, the passage of hydrogen through ATP synthase uses the exergonic flow of hydrogen ions to drive the phosphorylation of ATP. So this movement of hydrogen ions through the ATP synthase helps in the synthesis of ATP. Thus, the energy stored in a hydrogen gradient across a membrane couples the redox reactions of the electron transport chain to ATP synthesis. Now this is a very important sentence and a $1 million sentence, that the energy of oxidation reduction reactions when electron passes through different carriers, this energy is utilized to push hydrogen ions from mitochondrial matrix into the intermembrane space, and then when the hydrogen ions are in large concentration in the intermembrane space, they try to sneak back into the mitochondrial matrix, and in this way they uses the route provided by ATP synthase, and the ATP synthase synthesizes ATPs in this way. At this point, you may be wondering how the electron transport chain pumps hydrogen ions into the intermembrane space. So the energy, researchers have found that certain members of the electron transport chain accept and release protons along the electrons. So the aqueous solution inside and surrounding the cell are a ready source of hydrogen ions. At certain steps along the chain, electron transfer causes hydrogen to be taken up and released into the surrounding solution. In eukaryotic cells, the electron carriers are spatially arranged in the intermitochondrial membrane in such a way that hydrogen ion is accepted from the mitochondrial matrix and deposited in the intermembrane space. The hydrogen ion gradient that result is referred to as proton motive force, emphasizing the capacity of the gradient to perform work. So the hydrogen ions present in the intermembrane space, they are in bulk, they possess potential energy, and they can move into the mitochondrial matrix through concentration gradient, but they need a route which is provided by ATP synthase. The force drives hydrogen ions back across the membrane through the hydrogen channels provided by ATP synthase. In general terms, chemiosmosis is an energy coupling mechanism that uses energy stored in the form of an H ion gradient across a membrane to drive cellular work. So what is chemiosmosis? It is basically an energy coupling mechanism, and it uses the stored energy in the hydrogen ions which are present in the bulk in the intermembrane space. So this energy, this potential energy, is used to synthesize ATPs. In mitochondria, the energy for gradient formation means the energy for drawing the hydrogen ions from the mitochondrial matrix into the intermembrane space. This energy is provided by exergonic redox reactions along the electron transport chain, and ATP synthesis is the work performed. Now, chloroplast uses chemiosmosis to generate ATP during photosynthesis. In these organelles, light drives both electron flow down an electron transport chain and the resulting hydrogen gradient formation. Prokaryotes, as already mentioned, generate hydrogen gradients across their plasma membrane because they lack mitochondria. They then tap the proton motive force not only to make ATP inside the cell but also to rotate their flagella and to pump nutrients and waste products across the membrane. Because of its central importance to energy conversion in prokaryotes and eukaryotes, chemiosmosis has helped unify the study of bioenergetics. So it's a process which takes place both in prokaryotes and in eukaryotes. In eukaryotes, it takes place in the mitochondria. Prokaryotes lack mitochondria, so there this process occurs in the plasma membrane, and it not only results in the synthesis of ATP, but it also results in the movement of molecules and in the rotation of flagella. So now this is the summary of the whole process. FADH enters complex 2, NADH enters complex 1, and as these molecules deliver their electrons to a series of carriers, the exergonic energy is utilized to push these hydrogen ions into the intermembrane space, and as the hydrogen ions are present in bulk here, they have a proton motive force, they have potential energy, which helps them to sneak back into the mitochondrial matrix, and they find the route through ATP synthase, and they move inside through a process called chemiosmosis, and in this way ATP is generated. And during the lecture we talked about FADH2, which helps in the production of lesser number of ATPs. The reason is because it enters complex 2, and therefore when the process starts here, then lesser energy is given out for the pushing out of pushing out of hydrogen ions as compared to NADH, which enters complex 1. So the electrons delivered by NADH passes through more carriers as compared to the electrons delivered by FADH2. This will be the end of that. Thank you.